right here, as I recall. What we're looking at, the red sequence here, is our targeting sequence. That's the sequence that we put onto PCAP's plasmid. That is the 20 nucleotide sequence that we targeted. And that means that's the sequence that's going to be cut if CRISPR works, right? So that red box represents the sequence we put on the plasmid, the 20 nucleotide targeting sequence. On adenine 2 gene, next to that 20 nucleotide sequence, there has to be a PAM site in order for the Cas9 to cut. And in fact, there is a PAM site there, and I'll show you the actual sequence in a minute. When the 20 nucleotide targeting sequence in the single guide RNA directs the Cas9 to this position, it'll direct the Cas9 to cut where I've drawn the purple line. So Cas9 will cut there within that 20 nucleotide sequence. So think of that as step one. Just put in the Cas9 gene on that plasmid, and also the guide RNA on that plasmid. You put the plasmid into the E cell, that plasmid DNA will get to the nucleus, and the Cas9 gene will be transcribed and translated into the Cas9 protein. The guide RNA will be transcribed into the single guide RNA. The single guide RNA and the Cas9 will get together. They will travel to the adenine 2 gene, find it, find the place where it matches, this little 20 nucleotide sequence. I know that sounds amazing, but obviously it works. It'll find that 20 nucleotide sequence, and as long as there's a CAM next to it, the Cas9 will cut. So that's where the cut gets made. That's part one. That's the cutting part. Now we have to do the repair part. The repair part is assisted by this editing fragment that we put in. And we put in a lot of the editing fragment to try to push that this is the editing that occurs in our cells. We put in a lot of it. The editing fragment, looks a little short here compared to the rest of them. Okay. So the editing fragment, the first 50 base pairs match just to the left of where we plan to cut. So these 50 base pairs match these 50 base pairs on the adenine 2 gene. The last 50 base pairs match on the other side of the cut on the adenine 2 gene. And the 60 base pairs in between don't match at all. And so what happens is this red side will match up to the chromosome here, the green side will match up to the chromosome here, and this part won't match at all. This middle part that we designed, we put a BAMH1 side into it, and we put a stop codon into it. And I'll show you more details later about the positioning of that stop codon to make sure that it stops translation. Now, once this editing fragment finds the place where the cut is, it participates in that homology-directed repair of the chromosome. And remember, yeast really wants to fix this broken chromosome or it's dead, right? So the homology-directed repair enzymes get to work, and those repair enzymes include enzymes that do recombination. And in order to repair this break, the recombination enzymes will do a recombination event on that side of the break, and another recombination event somewhere over here on the other side of the break. Now, where exactly it occurs in this region doesn't matter. Where exactly it occurs in this region doesn't matter. It could occur there, it could be there, it could be there. This one could be here, 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 or here, right? As long as there's one on the left and one on the right, then the repair can occur. So it's the repair system that's doing that, okay? Now, once that recombination event has occurred, what it's done is connected this piece here through recombination to this, and then through recombination connected it back to here. And so maybe if you ever had a genetics class, you heard about crossing over. This is really what's happening here. So if you follow along this chromosome, you cross down, we pick up this thing, go around the loop, pick up this thing, come to here and cross back up, and now we're back with the chromosome. So now our chromosome has been repaired because it's intact, because now there's no break. There's no break. This little piece that's got the break in it just floats away and gets degraded. Okay? So now, if all that happens, we will have successfully inserted the 60 nucleotides in. On this side of the break, there's nothing different, right? Doesn't matter where this recombination event occurs, the sequence on this side is going to be the sequence of adenine 2. Because the sequence that we put in is a direct match for adenine 2. So even after the crossover, this is going to have the exact sequence of adenine 2 on this side, and the exact sequence of adenine 2 on this side. The only place that'll be different will be this position right here, where all of a sudden there's going to be this 60 base pair insertion. But other than that, when we sequence this later, we should get the exact sequence of adenine 2 to there, and then right at that point, boom, 60 nucleotide insertion, and then back to adenine 2. Okay? Make sense to everybody?
So the 20 nucleotide target guys Cas9 cut.
It's cut right here, right? So that 20 nucleotide targeting that we put onto Cas9 weeks ago, that's what that was for, right?
So we're putting in 60 base pairs in there. What's the highest you can actually reliably go?
Yeah, I don't know the answer to that, because what I do know is that as you go bigger and bigger, it becomes less efficient. So what the real limit is and what the practical limit is probably different, right? I know that hundreds can be done. If you want bigger than that, then you're going to have to make 2 cuts and do something a little fancier. Okay? Good thing break is coming. Yeah.
Where exactly does the Cas9 side relation pertain from? Is it directly in front of that NGG or?
No. Let me show you that in a bit. I'll show you that in another figure in a bit. So any other questions about this figure here before I talk about some other figures? Yeah.
If you're going to be over the base pair that you just discussed, and you're going to cut twice, how close together can those two be? Would it be kind of dangerous to put them too close together, where they need to be on different chromosomes or on the same chromosome just far apart?
You would, if you want to cut out a piece of a gene and replace that segment, they can be pretty close. I guess I don't know exactly how many, but I think 50 base pairs was enough difference.
Okay.
And it can be much farther. Okay? But I would definitely look that up before designing your experiment, right? Okay? There's a big literature out there now on CRISPR, and you can find all kinds of detail like that. Unfortunately, there's no one manual. Here's how to do CRISPR and all the details and all the cool things you'd like to know. You gotta kind of search around. Okay. Jennifer Dunn actually did a lab manual with some colleagues a bunch of years ago, but it's really horribly out of date now, unfortunately. Yeah.
Can you target multiple genes by putting
Multiple, yeah.
guide RNAs?
Yeah. So if you put in multiple guide RNAs, you can get Cas9 to cut in multiple segments. Yeah. Okay. Let's go to page 149, which is the start of experiment 5. Tracer sequence plus some of the CRISPR sequence, you know, what was shown in black in those other diagrams. The only thing missing are the 20 nucleotides that come at the very end, and so you put those right here. And so now, once you put in those 20 nucleotides, you would have a complete single guide RNA with the targeting sequence, and then all the rest of the sequence before that makes those loops and so on that the Cas9 binds to. So the only thing missing from a complete guide RNA here is putting in that 20 nucleotides, and you put that in right there. Okay?
you already explained why you need to be printed?
Because, Cas9 enzyme can only handle 20, it needs 20. If you do 19 or 21, works, but not as well. So for optimum efficiency, you want to do 20, yeah. Okay, the Cas9 gene is here that codes for the Cas9 enzyme, and it has, a yeast promoter here because we want this expressed in yeast. And this guide RNA, even though it's not shown, also has the yeast promoter over here, which they showed it, it's a sort of better diagram. Anyway, that's where you put the 20 nucleotides. Then there's some more information, and then this is the diagram that shows the non-homologous end joining and the HBr, and of course we did HBr. More information, and then this is the sequence of the actual primers that were used. Put the 20 nucleotides in, so 5' to 3' for both of them. These sequences are complementary to one another, and the 20 nucleotides inserted that are the target sequence are shown in yellow here. And so there's some more information on that. And then there's a diagram, the restriction-free cloning diagram that we talked about before, and then the protocol. So I just wanted to show you that all that information that was at the start of the experiment, we dealt with that, and it would probably be good to read that for some review at this point. Then at the end of the experiment, there are a bunch of useful diagrams. And now we're ready to take a look at these. So the first diagram shows the adenine 2 gene. This is from the yeast Saccharomyces cerevisiae. It's on page 173. Again, this is the yeast that's used in making beer, bread, and wine. And it's a really commonly used experimental organism, both in genetics and biochemistry. Really good model organism for eukaryotes. It's a single-cell eukaryote that can grow both as haploid and as diploid. So either one copy of the chromosomes or 2 copies. The cells that we just transformed and are doing our experiment on are haploid. So we're growing them as a haploid, and that means that you edit the chromosome, it's only got 1 copy, and so you will mutate the only copy of adenine 2 with those 42 caps, okay? If we were using diploid yeast, we'd have to get both copies on both homologs, and that's a lower probability. So remember that when you're dealing with organisms like mouse or human or whatever, you'll get some CRISPRs where you get one of the 2 homologs, right? And a fewer number where you'll get both, where both will get edited, right? And if you want to mutate a gene and create a mutation where the cell can only make the mutated copy, then you've got to get both copies, right? That's going to be a lower probability event, but they do happen. So I was talking earlier with folks about a mosquito experiment we did recently where we got, I think, 5 or 6 successful CRISPR lines, and 3 of the 5 had one of the 2 chromosomes CRISPR'd, and 2 of them had both chromosomes CRISPR'd. So, it's just something to keep in mind. Okay. So the adenine 2 gene sequence in yeast is shown here, and this is the entire adenine 2 gene. And so we're looking at just one of the 2 strands. We're looking at what we always refer to as the top strand. So this is the 5' end of the gene up here. This is the 3' end of the gene. If we were to show the promoter would be over here, upstream of this 5' end of the gene. What's in the gray box here is the part of the gene that's not translated. So this is the 5' UTR, the 5' untranslated region. So this region is transcribed, but it's not translated. So it's the 5' UTR. Translation starts at the start codon, and the start codon is this ATG right here. And I want to point out to you that that's not the first ATG. Without even trying, I can see another one right here, right? So there's an ATG right here. Cell knows that's not the start codon. It knows that one's the start codon. How does it know? Good question. But it knows. There are sequences that help the ribosome around the start codon, but we also know that the folding of the mRNA is really important. If the RNA misfolds, then you may not get translation starting at the right place, okay? So, anyway, there's the start codon. This is where translation starts, and everything gets translated to make the adenine 2 enzyme until you get down to here to the stop codon. This is TAA, which in the RNA would be UAA, you are awful, and that's the stop codon. If you divide this thing into triplets, going all the way down, triplet, 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 you'll see that none of the triplets are stop codons all the way through this one. There are no stop codons until you hit that one, okay? One way you can tell that you've got a gene is if you can go all the way through it and you see no stop codons, then that looks like a coding sequence, right? Because stop codons make up 3 of the 64 stop codons. So on average, you should hit a stop codon about once every 20 or 21 codons, right? So to have a long, long sequence like this, with lots and lots of codons and no stop codons, that's a gene, okay? Doesn't happen by accident.
This is actually a question from the Human Genome Project discussion, when they say they could sequence the 3.1 billion base pairs and know how many genes that was. Is that how they know what's a gene and what's not a gene?
Well, ask me again when I finish this, and we'll have another discussion because it's complicated. So we'll come back to that. What we're looking at here are the exons of the gene. We're not looking at the introns here. So this is the gene without any introns, right? So we've simplified here, okay? Now, at the end, after the stop codon, is another region that gets transcribed but not translated, and this is the 3' UTR. So the full transcript, the mRNA, would include everything from that T all the way down to that G, but only what's between the start and the stop codon gets translated, right? Okay. Now, the 20 nucleotides that we're targeting are the 20 nucleotides shown in yellow here. That's what we're targeting. And right next to that 20 nucleotide targeting is an NGG. We've got a PAM sequence right next to it. So remember, our sequence that we're targeting has to be 20 nucleotides, and then it must have a PAM sequence next to it. The PAM sequence is not part of the 20, but it's got to be right next to it. Okay? So the 20 nucleotides, that's the sequence that we put onto the CCAS plasmid, so that it would target this sequence in the adenine 2 gene. Okay? Everybody pretty comfortable with that? Okay, then we designed that 160 nucleotide long editing fragment, and 50 nucleotides of that editing fragment match this red sequence here, and 50 nucleotides match the blue sequence here, and then there were 60 nucleotides in the middle that don't match. So we're not seeing the editing fragment here, I'm just showing you where the editing fragment matches the adenine 2 gene. 50 nucleotides match here in the red, and 50 nucleotides match the blue here, and then there are 60 nucleotides in the middle of that 160 base pair fragment that don't match in the center. Okay? So that's just showing you where the homology is on the adenine 2 gene. Right?
The T after the PAM, why didn't you start the homology section there?
The what, sir?
The T after the PAM?
Oh, yeah, I think either me or somebody who was working with me made a boo-boo and missed that one. It turns out it's not necessary. We probably should have included it, but it works just fine. So yeah, we missed a key somehow. Or maybe when we asked the company to make it, whoever transcribed that in order and missed the key. And anyway, but it works fine. Not a problem. Okay? Any questions about this diagram? Oh, one last feature. This sequence here, grayed out and then in blue in this one, this is the location of the forward primer that we're gonna use for PCR later on. And this is the location of the reverse primer. Because later on, after we edit our yeast cells, we're gonna isolate DNA from these edited yeast cells, and we're gonna PCR amplify this region of the chromosome and sequence it so we can see if we successfully edited. Now, we're already gonna think we successfully edited, we're gonna pick pink colonies, right? And so we're gonna believe that we have the right sequence, but we're actually then gonna do the sequence to make sure that we did it the way that we expected. Now, in molecular biology, weird things can happen. We could get pink colonies and have an editing that we didn't expect. That could happen. And if anything weird like that happens, we'll see it because we're gonna do the sequence. Right? But CRISPR is remarkably accurate. So when we do these editing experiments, mostly you get exactly what you expect. So it's pretty reliable and pretty accurate in terms of the way it does them. Okay, so this was a complete sequence of adenine 2 showing various features that I think you'll find useful to remember. And then there's a bit of a key on here. And then on the next page, there's some hints about how to design the 20 nucleotide long guide RNA sequence. You can pick any sequence in the genome and hope for the best, but it's best if you follow some rules, because if you follow the rules, you can get more efficient CRISPR than you would if you just made it up randomly. So here are some of those rules, and I'm going to take you through these slowly, and we'll discuss each one a little bit. So the program that we use is a program called ChopChop. Thank you. It is a program designed to help you decide where you should design your targeted sequence, your 20 nucleotide sequence. And so what you tell ChopChop is, here's the sequence of my gene. You give it some other information. I want to use the enzyme Cas9. The gene sequence I'm using comes from the yeast Saccharomyces cerevisiae, and then it helps you to select the right 20 nucleotides. Now, it always gives you some choice, and so you're still going to have to pick something. And so here are the rules that you kind of want to follow. So an early region of the gene is preferred. Well, by early it just means really not at the end. So the problem with editing at the end of a gene is you might change then just the last few amino acids. This is the sequence that we want to target, this red sequence. That's going to be our 20 nucleotide target. So we're going to design a guide RNA that can hybridize to that target. What we want to do is target the antisense strand. Which one's the antisense strand? Well, the top strand here is the strand that has the same sequence as the messenger RNA, right? Done this before. So the bottom strand is your antisense strand. So you want to design your RNA so that it targets the bottom strand in our diagram here. So don't let the terminology fool you. People in this field, they use all this terminology. If they use plain English, it would be better. I've taken this from ChopChop so you know what you're up against. So that's why I'm taking the time to explain this for you so that you can understand what these things are. So you're going to target the antisense strand, which means you want to design your guide RNA so that when it gets transcribed, that sequence hybridizes for our bottom strand here. Okay? We can take a look at that later if you want to see how that works. Okay, we'll do that later. I'll have to do it on the board. Okay, so the antisense strand is preferred for targeting. That doesn't mean that if you target the other strand, it won't work. It appears to be more efficient if you target the antisense strand, and I have zero explanation for that. It's an observation. Base pairs, not 150, so at least I got it right here. And we're inserting a 60 base pair edit to the damage one site into the adenine 2, as well as this stop codon. The PCR primers we're going to use later are indicated here, and so I'll go through this part with you later on. I also have an entire sequence of the adenine 2 gene here, showing the translation, and then showing the location of those features again, for no particular reason other than you might like to see product actually looks like. So this includes only the coding. So this start codon here, and goes all the way to stop codon down there. Okay, so why don't we, so first let me see if there are questions about the CRISPR. So,